Thank you so much. So, hi everyone. So, good morning or good afternoon to the folks in Europe. So, thank you very much again for the kind introduction and for inviting me here to present my work. So, today my talk will be about my work that is done in the Bentley lab at the University of Maryland College Park. So, on creating this intranet of bio-nano things with the help of two things. So, the first would be electrogenetics, which involves engineering biosystems to be electronically controllable. And on top of that, with electrobiofabrication, which allows us to create living electrodes in order to integrate electronics with biological systems. So what is the Internet of Things? Actually, what is the Internet of Bio-Nano Things? So this idea is very evidently inspired by the recent trend of Internet of Things. And here I have a small clip that I believe, in a cute way, sums up this technology really well.
Zo, wat heb jij gemaakt? Laat eens even zien.
Not everyone is a master chef. We've got your back. McDonald's introduces the McDelivery Detector. The detector that automatically orders in when your food gets burned. How does it work?
We created the smoke detector in the iconic Big Mac shape. When smoke is detected, it will send your order to the nearest McDonald's restaurant. The detector is fully 3D printed. The inside is advanced hardware that uses the Internet of Things. And when it goes off, you hear the iconic McDonald's tune.
Turn bad luck into good times with this must-have gadget.
Okay, so a small disclaimer here is that unfortunately McDonald's did not sponsor our research. But nevertheless, in the same way as the McDelivery detector, Internet of Things are made up of very capable devices. They can detect, make decisions, and finally actuate certain useful functions. And Internet of Things enables these smart devices to communicate with each other and with other Internet-enabled devices. So while the advances in technology allows the connection between electronic devices, biological or living systems are still remain excluded from this vast electronics network. And we believe a major reason behind this is that they adopt very different communication modalities. So for example, for electronics, information is usually carried by freely moving electrons or electromagnetic waves. However, in living systems, information usually comes in the form of signaling molecules or ions. So they're unable to recognize and interpret the information sent by one another. Our group, however, proposed to use redox molecules and mechanisms to connect the two. So in biological systems, electrons were found to be transferred between redox active molecules. For example, here we have NAD plus NADH. These molecules can both receive and donate electrons, covering the electronic side of the communication. And okay, my apologies. I think my AirPod is not connecting properly, so I'm just gonna take them off. If you can still hear me, please give me a thumbs up. Okay. So now we have these redox active molecules that can, donate, receive electrons, which covers the electronic side of the communication. And also in both redox states, NADH and both NAD can interact with biological components such as the enzyme ethyl dehydrogenase here to pass on biological information. So we believe the dual feature of these redox active molecules is the key to bridge the two realms of electronics and biology. So by harnessing the dual feature of the redox molecules, the Bentley group, our group, first realized the concept of electrogenetics, where gene expression can be directly influenced and controlled with electricity. So the first system we developed was developed by rewiring the oxidative stress regulon SOX-RS that is native in a lot of bacteria, including E. coli, together paired with a redox active metabolite piocyanin and a redox mediator ferrocyanide. An electronic CRISPR-based transcriptional regulation system, I know it's very long, so we named it eCRISPR to be short, was also built on the basis of the SOX-RS system. And recently, our group introduced a new electrogenetic system that is based on the OxyRS regulon. So this system uses electro-generated hydrogen peroxide, or sometimes I'll just refer to it as peroxide, to turn on the rewired OxyRS regulon. So this regulon OxyRS is another primary oxidative stress regulon native to E. coli and is homologous, can be found in a lot of bacteria. The transcriptional regulator OxyR can sense peroxide and can be oxidized by hydrogen peroxide, and after that, its conformation will subsequently change to initiate the OxyS promoter. So compared to the previous SOX system, this current oxy-based system enables direct electroinduction, does not require additional mediators, and it is inducible in aerobic conditions. So in this work, we use hydrogen peroxide as the redox bridge to help transform the electronic signal into chemical and then finally into biological signals. This eventually allows us to achieve electronic and even automated feedback control of both protein activity, cell function, and even cell consortia behavior. So in the later slides, you'll see the numbers labeled on the top right corner to help you keep track of which biological systems the slide is currently referring to. So to start off, we have previously found that electro-generated hydrogen peroxide, as shown as this modeling data here, is very highly localized at the electrode surface. And in order to ensure efficient signal transfer, we use electrobiofabrication to assemble enzymes and cells onto electrodes. So for enzyme assembly, we use mediator iridium IR to facilitate the oxidation of gelatin and horseradish peroxidase, or HRP, together to form a hydrogel. And for cell assembly, as shown on the bottom, we use the mediator ferrocene FC to facilitate the oxidation of the thio groups that is present on the ends of the thiolated PEG monomers for it to cross-link and then at the same time entrap E. coli within the gel, forming a biomimetic artificial biofilm. So here we showed that electrobiofabrication is highly customizable. So first, you can easily program the hydrogel's area and shape simply through changing the electrode's design. So here for both HRP and E. coli assembly, we can see that their distribution is homogeneous and clearly defined by the electrode, and also their biological function, which namely is HRP's enzyme activity or the growth-related quorum sensing activity for the E. coli, are both shown to be preserved and can be spatially configured. And furthermore, you can easily control the thickness of the artificial biofilm by varying the time of electrodeposition. We showed that here when you apply, the longer you apply voltage to cross-link, the thicker you can get you can make the artificial biofilm. And for electrode choice, we later transitioned to the clear ITO electrodes for optical assessments. So in this slide, we presented a 3D printed ITO-based device that is designed and fabricated by my collaborator, Dr. Chenyu Chen, in our group. So with this ITO device, we can both apply an oxidative voltage to assemble proteins or cells, or you can apply a reducing voltage to generate peroxide through this oxygen reduction
for the
reaction
serological
OR.
pipette.
So
The
on the
long
right,
glass
you
ones
can
should be under
see
here.
both the profile of the current
Voila.
or the profile of the charge and the generated peroxide during this reaction. We found that the peroxide level exhibits linear dependence with duration and charge.
Oh, got it.
So with the interface and the hardware developed, we moved on to the interesting part where we create an OxyRS-based eCRISPR transcriptional regulation system to demonstrate electronic control of cell functions. So apart from gene editing, CRISPR technologies can be used to easily control gene expression as well with techniques like CRISPR activation, CRISPR interference for repression, and even multiplex control of several genes at the same time. So starting off, to achieve CRISPR activation, we place the guide RNA SG108 downstream of the OxyS promoter. So upon sensing peroxide, the guide RNA will be transcribed and form a complex with the constitutively expressed dCas9 omega. The dCas9 omega, suggested by its name, is the deactivated version of the endonuclease Cas9 that is fused with the omega subunit of E. coli's RNA polymerase. So this SG108 dCas9 complex will be ultimately directed to the upstream of the gene of interest to initiate its transcription. So on the right-hand side, we showed that the expression of the guide RNA, which is shown on the top, and also the gene of interest GFP shown on the bottom, are both inducible, can both be induced by hydrogen peroxide. So next, we wanted to demonstrate eCRISPR activation with actual electro-generated hydrogen peroxide. So on the left-hand side, we first showed that the general workflow of all the experiments that are done on our ITO device. So first, we electroassemble the cells by mixing the cells with the monomer, apply some oxidative voltage, and then it will cross-link into this hydrogel film. And after ample washing, we added growth media into the wells to submerge the artificial biofilm. And at the very end, we will apply a reducing voltage to generate hydrogen peroxide for electroinduction. So after induction, we found that up to 70% of the electro-induced eCRISPR cells express GFP with 30 minutes of electroinduction. And remarkably, the fluorescing fraction increases with charge applied to the cells. So now that we've shown that we can control the single cell behavior with eCRISPR, we wish to expand toward controlling population behavior and hoping to translate the electronic signal, which is hydrogen peroxide, into biological communication signals by tapping into bacteria's own way of communication, that is quorum sensing. So I like to generalize quorum sensing as how bacteria talk to each other. But by secreting these signaling, but instead of actual, using sound waves, but they talk by secreting these signaling molecules that are called autoinducers. So quorum sensing bacteria have cognate receptors for recognizing autoinducers to turn on various genes that govern communal behavior like luminescing or biofilm formation. And there are two major quorum sensing systems that is characterized by the autoinducers that they secrete. So the first group of quorum sensing bacteria secretes autoinducer 1 or AI1. AI1s are usually species specific, where different bacteria can make and recognize a different and specific type of acylhomoserine lactone with a different R group. And the second group secretes and recognizes AI2. And opposed to AI1, which is super species specific, AI2 is a universal language across multiple species, including E. coli. And instead of being a derivative of an acylhomoserine lactone, AI2 has a unique boron-containing cyclic structure. So in the following slides, we will show how we use the eCRISPR system to modulate E. coli's quorum sensing communication to control the behavior of microbial communities. So to begin with, here we will show that with the help of eCRISPR activation, we can make E. coli speak a foreign language that is originally native to Pseudomonas aeruginosa, or namely to make E. coli secrete Pseudomonas AI1, opening new lines of communication. So for this, we swapped out the genes of interest from GFP to LAS-I here, which is the enzyme responsible for AI1 production. Our results on the right showed that AI1 levels were both peroxide inducible in liquid culture and also directly electrically inducible on the ITO device. And here on the most right-hand side, we presented a co-culture setup to demonstrate cell consortia control. So after electroinduction, our results showed that the planktonic culture can successfully receive the AI1 secreted from the electrode-bound eCRISPR cells and then respond with a higher level of fluorescence. And next, thanks to the versatility of CRISPR, here we show that using CRISPR interference or CRISPR-I to inhibit E. coli from speaking its mother tongue, or which is the AI2 quorum sensing system. To achieve eCRISPR-I, we designed the guide RNA LuxS1 that is complementary to LuxS, which is the terminal production enzyme of AI2. So after confirming its efficacy, our results also showed that we can turn off AI2 signaling both through peroxide induction and also through direct electroinduction. And finally, we combine the previous eCRISPR activation of LAS-I and eCRISPR inhibition of LUXS to create a bilingual strain that, upon sensing the electronic cue, changes its spoken language from AI2 to AI1. So on the right, our results again showed that either through peroxide induction or electroinduction, the induced cells displayed higher AI1 and lower AI2 levels. And we also noticed that, interestingly, the cells that received more doses of peroxide or more pulses of the electroinduction made even more AI1 and even less AI2. And these results hinted to us that OxyRS eCRISPR could potentially be dynamically regulated. So encouraged by these results, in the following slides, we will shift gears and try to demonstrate redox-mediated dynamic control of both enzyme and eCRISPR activity. So here we'll start off by showcasing how we regulate HRP activity, or horseradish peroxidases activity, through controlling the generation of its substrate hydrogen peroxide. So for this, we redesigned the previous ITO electrode, and now for it to boast two interdigitated electrodes. So on this device, you can apply voltage alone on the bare ITO working electrode 1, or as labeled WE1, to generate hydrogen peroxide as writing data. Or you can apply voltage alone on the working electrode 2, where horseradish peroxidase is pre-assembled on with the hydrogel gelatin to detect the generated peroxide through HRP catalyzed reaction, and then record said data by generating a current output. So to see what it actually looks like, on the right, we repeated this process of producing peroxide on working electrode 1 for 1 minute, as you can see from the dark blue curve. So with the increasing charge, it means that we're currently making peroxide. And then we detect the hydrogen peroxide on working electrode 2, where we plotted the current that obtained on working electrode 2 with the pink curve. And at the 2-minute mark, we recorded the endpoint current, which is shown by this darker pink circle. So after repeating this process multiple times, we saw an increase in the working electrode 2's current, which is the dark pink circles, every time working electrode 1 makes more peroxide. This increased HRP activity on working electrode 2 also suggests that this platform can definitely write and then retrieve the written information in a manner that is analogous to computers' random access memory. And moreover, our device can also store and then retrieve the information that is written on the working electrode 1 and read that information at a later time. So what does that mean? So what actually shows is that for this, we first apply the voltage to generate hydrogen peroxide on working electrode 1. This time we generate for 10 minutes for a little bit longer. And then we apply 0 volts on working electrode 2 to acquire the output current, as shown at the bottom. So similar to the previous slides, we saw that the current stabilizes around the 2-minute mark and then stays a similar level for the entire data acquisition period for around 50 minutes. And this shows that our written information can be properly stored in our HRP device. And also, immediately after the first data acquisition period, we apply a second round of charge input on working electrode 1 to generate more peroxide. And also, similar to the previous slide, we saw this increase in the output current when we start recording the current on working electrode 2. So this once more indicates that our device is capable of retrieving its own written information. So moving on from this slide, we will show how we realized automated dynamic control instead of the HRP's enzyme activity. Now we show that we can dynamically control OxyRS-based eCRISPR as a dynamic control of cell functions. So first, we proved that the OxyRS induction system can be administered repeatedly, as shown by these 3 repeated inductions. So to be more specific, we used a stripped down version of the OxyRS CRISPR genetics circuit, which no longer contains the CRISPR parts, but right here we just put like a degradation tagged SFGFP under the OxyS promoter. So this is just for demonstration. So later we'll still show that with the OxyRS eCRISPR, we can also dynamically regulate that as well. So as this demonstration, after we carried out the first peroxide induction, which is shown on the top panel, we saw that the induced cells, which is the blue and the red bars, displayed higher fluorescence compared to the uninduced cells. We then introduced the cells to a second round of peroxide, and initially we saw this inducible behavior where the cells that received more hydrogen peroxide, a higher level of hydrogen peroxide, displayed higher fluorescence. This phenomenon, however, quickly disappears after 30 to 45 minutes. This similar behavior of the cells having this inducible behavior and then quickly disappearing can also be observed when we induce the cells with a third round of hydrogen peroxide. We think these results showed us that peroxide induction with the OxyRS induction system is transient, so therefore can be administered repeatedly and most importantly have the potential to be dynamically regulated. So inspired by these results, we then built a full bioelectronic system where we called BioSpark to streamline the communication from electro to bio and then back to electronics. So BioSpark is this device, a platform, integrated three major components. So the first one is the potentiostat as the electrochemical instrument. And then we have a fluorescence module to pick up emitted fluorescence from eCRISPR cells as the bio to electronic signaling output modality. And then finally, we have an environmental chamber to house the ITO device with the cells on top for incubation. So initially, we used the BioSpark to characterize the on-electrode cell growth and also the on-electrode GFP expression dynamic. So here we found that after electroinduction, which is indicated by the yellow zone, we see that the GFP level quickly goes up and then eventually tails off. A custom algorithm was developed by us to help us track the expression dynamic using the ratio of the slopes in the curve. And since this is a control-focused seminar series, I finally have the chance to go over our rather simple yet pretty effective algorithm. So our algorithm calculates the slope between two adjacent fluorescent values and tracks and updates the maximum slope along the experiment. So for every recorded slope, the algorithm updates which slope is the maximum slope and then computes the ratio between the slope and with the maximum slope. So when two consecutive slope ratios fall below the user-defined ratio threshold, our algorithm will deem the fluorescence level has now reached a plateau. So here we can finally see the BioSpark and the eCRISPR cells and also the algorithm in action to demonstrate the first fully and direct electronic control of gene expression. So our automation program starts out by giving the electrode assembled eCRISPR cells the initial electroinduction, and then the fluorescence module will then picks up and digitizes the optical signal to be processed by our algorithm. As explained in the previous slide, our algorithm can effectively identify the plateaus in the fluorescence profile with the ratio threshold metrics we predefined. And when the plateau is detected, the algorithm will then prompt the built-in potentiostat to apply a second round of electroinduction. And this process will repeat on and on until the fluorescence reaches its target level and the experiment will end. So apart from tracking the fluorescence optical signal, our closed-loop BioSpark system has the potential to receive and process electronic signals for feedback control as well. So one example will be tracking the electronic current generated during electroinduction as a proxy or indication of cellular respiratory activity. So here on the left, we present the current profile during every round of electroinduction. So again, on the top graph is the is the fluorescence graph that you've seen in the previous slide, and then the yellow zone indicates when the electroinduction is taking place. And here we label them as accordingly to its own current profile. So actually during the electroinduction period, we have to supplement oxygen to all the samples to counteract the cell's respiratory activity, which is actively depleting the oxygen at the electrode surface. We found that even with supplementing external oxygen, there is still a decrease in the current during electroinduction, which indicates that the cells are still actively metabolizing and using up the oxygen that we supplied. This electrochemical information of how low the current gets, or how long does it take to reach that certain amount of charge for electroinduction, can potentially be used to track the cell's general metabolic activity. And also on top of this, we can engineer cells to produce redox active metabolites like shown here, phenazines, which is a class of secondary metabolites secreted by bacteria like Pseudomonas aeruginosa. So certain phenazines that are more popular, more well-known, for example, is piocyanin shown here, and also the phenazine carboxylic acid, which is this precursor. So piocyanin is actually a toxin secreted by Pseudomonas aeruginosa to eliminate its potential competitors. These redox active metabolites can be read through simple electrochemical techniques like here. Shown here, we use differential pulse photometry, which is a rather sensitive electrochemistry method to detect these redox active metabolites. We showed here that with increasing amount of PCA, you get an increased current. So potentially this could help us turn potential biological information into a electronic information back to be feedback into our control system. And these electrochemical outputs can potentially replace the optical output that we're currently using now, which is fluorescence, for multimodal input and output controlling schemes. So ultimately, our goal is to connect different biological systems and even with human users to achieve supervised cascaded control. So here as a demonstration, we're linking both the BioSpark for regulating eCRISPR functions in cells and also the HRP platform for regulating enzyme activity, two seemingly disparate systems together to form a network via the internet. Specifically, we are using the HRP platform to feedback control the eCRISPR-mediated expression in BioSpark. We also purposely placed the HRP platform in a separate room in a remote location to mimic the Internet of Things and showcase wireless remote control. So here on the right is the full system diagram for the entire network that will be explained more in detail in the next slide. So how it works is we start off in the local BioSpark, and then you can follow along which system is in action with the little illustration on the left. So we start off by applying the initial electroinduction to the eCRISPR cells in the BioSpark. And as the GFP expression reaches a plateau that is detected by BioSpark's algorithm, this time, BioSpark sends a message to the remote HRP platform to check if more electroinduction is needed. So the HRP platform goes through a preset of program of generating peroxide and then the retrieval of the output current. So in the beginning, the current is low. The output current is low and it's below that current threshold. And the status of the low HRP activity is relayed back to the local BioSpark, which we programmed for the BioSpark to recognize this message and then initiate another round of electroinduction. So the entire process is again repeated until the fourth checking by the HRP platform. So this time, this current exceeds our preset current threshold. This status will then cue the HRP platform to send a confirmation text to your phone asking if you would like to terminate this experiment. By responding with a yes, the local BioSpark will then initiate its termination process, which here we use photo bleaching of the GFP as a demonstration of destroying the expressed product. So at the end here, you can see that the fluorescence drops from the photo bleaching. We would also like to notice that despite the similarity to the results in the previous slides, this is a separate experiment and additionally shows our system is very robust and highly reproducible. So in conclusion, in this work, we enlisted electrobiofabrication's help to build a living electrode that provides a communication interface. And by using peroxide as the redox bridge, we achieved real-time control of enzyme activity. And also we engineered an oxy-eCRISPR system for influencing consortia behavior and achieve closed-loop control of gene expression. And finally, we coupled redox and wireless electronic communication to network seemingly disparate biological systems for supervised and cascaded control. So finally, I would like to thank my funding sources, Bentley Labs funding sources, everyone in the Bentley Group, Payne Group, and the Fischl Institute. And last but not least, thank you so much for coming to my talk, and I will take any questions.